All right, back with the final part of our sequence alignment lecture. So I'm going to be talking all about substitution matrices. And we've seen a couple of examples of this, well, just one really, with that eDNA full matrix. I kind of glossed over it a little bit because I knew we would be talking about it in more detail in this part of the lecture. So, as I mentioned, DNA and protein matrices are slightly different. Protein matrices are often or scoring matrices. Sorry, I keep saying matrices. That's not very clear. I apologize. Protein scoring matrices are often a lot more complex since we have to take into account amino acid families. So, if you remember back to your biochemistry classes, there are certain types of amino acids that can be grouped together in what we would call a family, and that's all related to their side chain properties. So we can have hydrophobic amino acids, we can have hydrophilic amino acids, we can have side chains that are charged, uncharged, we can have all sorts of different things associated with our amino acids. And lots of research has been done into identifying what kinds of amino acid families are more likely to replace other amino acids when it comes to things like genetic mutations and things like that. There are some amino acids that we know have to be there for certain protein substructures to be formed. So we're able to add a really big penalty in our protein scoring matrices if those amino acids are substituted, and so on and so forth. The reason that we can do this is just through refining our match and our mismatch scores. So we can provide different values for different amino acids if they're mismatching together, dependent on how likely they are going to be successful based on their amino acid family, and then also what we know from previous research. So they're very, very large and a lot more complex than what we see with the DNA scoring matrix. So a DNA scoring matrix is usually fairly simple. Down the diagonal, we would see the score for a match, which is usually going to be some value like 1. Could be some other positive value as well, depending on how we want to slice and dice it. And then our mismatches are all these off diagonal values. So in this case, for this protein or DNA scoring matrix, our A's swapped with T's or C's are penalized at a -5, and our A's swapped with G's are penalized at a -1. And so if you remember, A's and G's are kind of a similar shaped nucleic acid, whereas the T's and C's are also a similar shaped nucleic acid. So this is a more complicated DNA scoring matrix than we've seen before because it's taking that into account. They don't always, but that's why we see those different values here. A lot of our previous examples we've done in this lecture, we'd have the same value all along the diagonal and then the same value for mismatches as in these upper and lower triangles. But they can really be whatever values make sense for the biological question at hand. We typically in bioinformatics use 2 kinds of protein scoring matrices. So one's called PAM, which stands for point associated mutation, and the other one's called BLOSSOM, which stands for blocks substitution matrices. Now, PAMs are based on these things called accepted point mutations. And this is when the substitution of one amino acid with another actually persists in the sequence. So the idea behind this is that if the mutation was causing harm to that protein, it would not likely become a predominant mutation within that gene pool because it would be detrimental to the organism or the cell or the whatever we are dealing with here. So how this was created was way back in 1978 by Margaret Dayhoff. She collected data on how often amino acids changed from one amino acid into another amino acid. And she did this for every single amino acid that we have. She recorded it specifically in sequences where 1% of the amino acids had changed. The data was only applied to sequences that were 1% different. So this was called the PAM1 matrix. Scoring matrices that are derived from that are applied to more divergent sequences are just calculated by multiplying the PAM1 matrix by itself a number of times. So PAM1 is for really similar sequences that are 99% similar. And as you multiply that out, we'll get into things like PAM250, which are for sequences that are more divergent. So here's an example of the PAM 250 matrix. So this uses the single letter codes for all of our amino acids and goes through and says, here's the match value. So you'll see even the match value is not consistent for each amino acid. And then all of the different mismatch values for that amino acid. And if we look at things, let's find some, there's certain ones in here that are like, oh, cytosine is usually always replaced by or matched up with the cytosine. It's very important in a lot of our protein substructures. So we have a really high number associated with that. Same here with tryptophan. There's not a lot of amino acids that are shaped like tryptophan that fill up that much space. So if we have a tryptophan, we really typically see it's associated with another tryptophan in a different sequence. And those penalties for mismatches are a lot higher than what we see in other cases. So PAM 250 is useful even when proteins share approximately 20% identity or similarity. So really getting further out in our divergence. Blossom matrices are a little bit opposite from the PAM matrices. So they were determined in a similar manner, but in a different form. A different form when we look at the PAM matrices. So the matrix multiplication was not used to account for evolutionary distance in Blossom. The numbers that we'll see at the end of the Blossom, so like Blossom 62, etc., are more clearly related to evolutionary distance. Blossom 62 is actually the default scoring matrix used by many tools, and it represents a 62% identity between sequences. So effectively, the PAM and the Blossom numbers work in opposite directions. Lower numbers for PAM are more similar. Lower numbers for Blossom are more dissimilar, and then vice versa for the higher numbers. This is a good example here of just how that relates to when a sequence is less divergent and more divergent. Oftentimes, we're going to be sitting somewhere in the middle with this Blossom 62 as a starting point because we typically don't know how divergent our sequences are. And then we'll fine-tune things as we get a clearer answer on that based on what we see from the results. There is a little bit of a twilight zone. So after a certain number of mutations between homologs, it becomes too difficult to recognize the homology at the sequence level. You might be asking yourself, where does this happen? How can we be confident that similarity and alignment are telling us sequences are homologs? So here's the math behind this twilight zone. When a protein undergoes 250 changes per 100 amino acids, it may still have about 20% sequence identity with the original protein. This is because the same amino acids will change many times, or amino acids will revert back to what they were earlier. The 20% identity can still often be recognized as significantly related or homologous, but there are no guarantees from percent identity alone. Usually the threshold that we use as bioinformatician is a 40% amino acid identity as a good threshold to conclude that two proteins are homologous. When there is 360 changes per 100 amino acids, we would end up getting about 15% sequence identity, and the 50% identity is not enough to conclude there's any homology from a pairwise comparison. So two proteins that are completely unrelated just by random chance can share between 10 to 20% sequence identity. So if you're in an exam and I'm asking you, these two sequences have 15% sequence identity, are they likely homologs? Wow. The answer is no, because of this math that we have here that someone else has figured out for us and published in a research paper. So PAM 250 captures that point of exactly 250 mutations per 100 amino acids, which is right in that twilight zone, the area, the furthest that we can go kind of to recognize homology from sequence alone. In our next week lecture, we will be talking about another statistical test called the E-value that can also help us assess confidence when it comes to sequence identity and homology. If you want additional readings, there's 2 textbooks that you can look at in the library, and those are the relevant chapters. Otherwise, you can also reach out on the discussion board, and I'm happy to clarify. Hope everybody has a great week. This is the end of our parts, and we'll catch y'all next week.